Hello, welcome. Welcome to this year's Ribicoff Lecture. This year's Ribicoff Lecturer is Dr. Pat Sullivan, Distinguished Professor in the Departments of Genetics and Psychiatry at University of North Carolina at Chapel Hill, and Professor in the Department of Medical Epidemiology and Biostats at the Karolinska Institute in Stockholm, Sweden. He received his MD at UCSF and completed psychiatry residency at University of Pittsburgh. He then moved to New Zealand. After 5 years there, he was recruited back to the US in '96 to work with Ken Kendler at Virginia Commonwealth. In 2003, he moved to the Department of Genetics at UNC Chapel Hill and now splits his time between UNC and Karolinska. Key among his many research accomplishments is that Dr. Sullivan is founding member, lead PI, and coordinating committee chair of the Psychiatric Genomics Consortium, the PGC, a collaboration that played the major role in revolutionizing our understanding of the genetics of psychiatric traits. For many years, he fought against a tide in the research community where it had become fashionable for a time to argue that GWAS approaches somehow wouldn't work for complex genetic traits, and especially wouldn't work for psychiatric traits. Of course, this isn't true, we know now, and no one has contributed to that state of knowledge more than Dr. Sullivan. The goals of the PGC, Psychiatric Genomics Consortium, are to conduct GWAS by means of large mega-analysis for ADHD, autism, bipolar disorder, major depressive disorder, and schizophrenia, and now also substance use disorders, eating disorders, and anxiety disorders, and this list continues to grow. Dr. Sullivan has been lead author on numerous of their most important articles. There are too many to list, but a recent one was the Nature Genetics paper identifying the 44 major depression risk loci, which is something really they said it couldn't be done. But it could, and it took a cast of thousands, and Dr. Sullivan was a key leader in the effort. So a couple of brief words about the Ribicoff series. This is named for Abraham Ribicoff, who was a Connecticut-based politician who served in state roles as governor and in national roles as US Senator for several terms and in the House of Representatives for Secretary of HEW for the younger folk among us. That's housing, health, education, and welfare. And he was a visionary with regard to psychiatry research. And here's a quote, and thanks to Bob Mallison for finding it. "Let us evaluate our thinking about mental illness and how we've dealt with it. Let us ask ourselves, are even the modest advances we've made in recent years on the right track? Or do we need an entirely new point of departure? Are we exploring every possibility in innovation, psychological, social, and biological?" And look at the data on that. That's from 1961. We have had a very distinguished list of past Ribicoff lecturers. It's a great pleasure to welcome Pat Sullivan as the 2019 lecturer.
Cool. Thank you very much. Good morning, everyone. Let me see if I can get my talk up without problems. Excellent. And thank you for the exceptionally kind introduction. I hope to do well by you in this talk. And I also appreciate the invitation and also the kind hospitality you've shown so far. Today I'm going to talk about this, and the title breaks neatly into an outline. I'll first discuss the genomic findings we have in psychiatry to date, the diagnostic implications and therapeutic implications. A bit of background to begin. So does anyone recall the lesson of the pink spot? This was back in the 60s when people found that if you took the urine from people with schizophrenia, put it on liquid chromatography, they had this pink spot that was, had excitement because it implied that there might be a metabolite that was identifiable. There was a literature on the use of hemodialysis in psychosis, where, and it's an appealing notion, you've got somebody who's psychotic, you do dialysis to get out small molecules, and they get better. This was a paper in Nature in 2018 talking about a linkage study finding a locus for schizophrenia in chromosome 5. And as one of my senior human genetics colleagues, who shall remain unnamed, 15 years ago told me that schizophrenia was like cancer. There was literally no way we were ever going to crack its genetics because it's fundamentally too heterogeneous. A couple of quotes for people that were into rare variation from Mary Claire King and John McClellan and David Goldstein, basically saying that really what schizophrenia is all about is the rare variant of strong effect ballpark. And we have empirical data right now for all of these to suggest that they aren't true. So the lessons of the pink spot went away when people understood that it was just an artifact or some treatment-related issue. The hemodialysis finding went away when they did the double-blind study. Oops. The Nature paper from linkage for chromosome 5 was reputed within a year when it came out. And I would argue that a key part of my talk is that statement is incorrect about cancer. And we've done enough rare variant stuff right now that I'll talk about to indicate that, in fact, that probably is not the case. So what's going on here? And effectively what it is what Ken Kendler called the great professor principle. We're in a field where the tradition has been that there's an influencer out there, somebody that everybody respects, that's super smart, scary smart, and they tell us the way reality is, and we go with it. And usually that's been because we're in an uncertain situation without fundamental data, and then basically the opinion exceeds data. But then over time, opinion becomes the reality. And I think we need to be conscious of that in this field in particular. Moreover, we've all had issues, all areas of medicine, as I think has been reasonably well established right now, has had issues with reproducibility. There are a lot of findings out there that simply do not hold up over time. Or if the reasons for this are complicated, and that's an entire separate lecture, but clearly reproducibility has been an issue. And in the last decade, 15 years probably, my field, cytogenetics, has gone from people coming out there and saying, hey, look at this cool thing. I've got a soft hit, but the gene is incredibly cool, to basically, we love the null hypothesis. In other words, we will take a finding and we will go after it for a year and a half. And if it doesn't go away, then we'll start to believe in it. And I think that switch in attitude has actually been incredibly important. Now, to be truly fair, though, all of these efforts that I've talked about have been from people who were passionate, smart people, and they were really trying to do a good job. I would also note historically that every generation of psychiatrists has taken its crack at schizophrenia. And basically, a lot of the techniques that have worked really, really well in other areas of medicine have not worked in psychiatry, or not very well. And again, the reproducibility crisis is a thing. And I would argue, and I hope I can convince you in this talk, that the data that suggest right now that we are clearly making progress. We know what we need to do, but the results are not what anyone would have anticipated 10 years ago. And that's really kind of a key issue. Okay, to talk about genetics, just in general terms, basically it's been our major clue for the etiology of just about every psychiatric disorder. Studies go back 70 years or more. And the Greeks even presaged much of this with their studies or clinical observations of hereditary madness, for instance. And for schizophrenia, as one example, really the situation now is that the risk is probabilistic and not deterministic. Family history has been shown in generations of studies now. That the relative risk of schizophrenia, if a person has schizophrenia, the risk in their relatives is about tenfold. That's been relatively widely replicated. The finding is extremely strong, well reproduced. However, the absolute risk only goes from around 1% to around 10%. The vast majority of people with a family history of schizophrenia schizophrenia tends to run in family just once. To find multiplex families is a little bit odd. The twin heritability is 65%. That tells us right there that environment plays about a third of it is environmental, 2/3 genetic. Moreover, the one particularly obvious indication of this is if you look at the pattern of monozygotic twins. If one more or less genetically identical twin has schizophrenia, the risk to the co-twin is about 50-50. So schizophrenia runs in families, but not strongly. There's been a lot of look for convincing single gene causes, and I can talk about this in the question area, but I haven't seen anything that convinces me of that. Moreover, I think this argues that schizophrenia will tend to be a subtle process. Moreover, if you think about it, even when a person is at their most severely ill, it's surprising how much of the fundamental neurology is actually intact. Sensation is fine, motor is fine. Certainly there are impacts on cognition and memory, but you can have a conversation with somebody, even when they are at their most severely ill. and it's an episodic course. It's not as if one given onset things become irrevocably worse. And many people with schizophrenia have some subtle abnormalities that are detected premorbid, but many actually have a developmental trajectory which is completely within normal limits. So again, I think these factors argue for a subtle process. Now, a data slide looking at rare genetic variation for schizophrenia. This is a relatively up-to-date set of findings. Basically, it's this. We have 5 different disorders here: autism, developmental delay, epilepsy, intellectual disability, and schizophrenia. Here we have the p-value, and this is looking at protein coding variation that piles up in genes. Each of these points is a gene with its associated p-value from that analysis for this disorder. And as you can see, autism, there's a ton of stuff. Developmental delay, a ton of stuff. A few things for epilepsy, a lot for intellectual disability, and basically just 1 for schizophrenia. This is SETD1A, which has been published. It's also a developmental delay gene, but it probably is a schizophrenia risk gene as well. But this variant is present in 0.1% of cases and basically 0% of controls. So it's got a risk, but it's certainly something that isn't likely to apply to many, many clinical populations. There's a study right now called Schema, and if you actually do a web search, they just made their results public on the web, off the Broad. Basically, this is extremely well done schizophrenia exome mega-analysis, 23,000 cases, and they find 3, maybe 6 genes. And this is notable because in context, autism actually has something on the order of 102 genes at about half the sample size. And there's also some findings that are emerging for OCD and Tourette's, which are pretty cool. But again, you get the sense that the pattern for schizophrenia and for adult onset disorders may be quite different than disorders that have their onset, that are severe and have their onset very, very early in childhood. Now, copy number variation. Generally, we inherit for the autosomes 2 copies, 1 from mom, 1 from dad, of every genomic segment. However, there are places in the genome that are predisposed because of the local structure for that not to occur. These are copy number variants. So, for example, 22q11 deletion syndrome, DiGeorge syndrome, velocardiofacial syndrome, et cetera, is characterized by a 3 million base pair segment where people have 1 copy instead of the normal 2. The 50 to 60 genes in that area have half the dose than they usually have. And this region has been widely replicated across the world to increase risk for multiple neuropsychiatric disorders. And so autism, schizophrenia, epilepsy, ID, et cetera. And this is the large study from the PGC, and taken together, about 10 of these have been identified. They're all most of them are multigenic. In other words, the number of genes within the copy number variant region varies from 3 up to 50. So it's not like it's a clean single hit. There are a couple which do hit single genes, Nerexin 1, top 3 beta. Most of these are de novo. In other words, the parents don't have them, but they occur as mutation in the making of that child. And there probably certainly are more that are out there, but our technology is insufficient at this point in time. The technologies that we've applied are insufficient to find them. We'll get more with better tech and larger studies. And so altogether, something on the order of 2 to 3% of people with schizophrenia will have one of these. And for autism, it's probably in the order of 10%. Typically, the more severe, the more you're going to find. In terms of common variation, so we've gone from rare exonic variation to rare copy number variation. This is common variation. And what I'm showing here is a graph of what we learned over in the whole field, all of complex trait genetics. There's been over 3,000 studies that that have applied genome-wide association. And there's more now actually. This was just the number I got from about 6 months ago. Basically, these have studied over 2,500 diseases, traits, and lab measures. And there's about 32,000 SNP trait, single nucleotide polymorphism trait, or SNP trait associations that are significant. Many of these are actually quite strong, or subset of these are quite strong. Odds ratios greater than 5, but they're relatively easy things. Infectious disease, rare adverse drug reactions. Again, if I could do this again, I would certainly study eye diseases. They're awesome. They have like tons of small things. They're easy. You take a photograph of the retina and you've got your diagnosis. They're great. Or things related to human pigmentation, hair color, skin color, et cetera. There's a small handful actually that are super strong, odds ratios greater than 10. But as you can see, these are basically not psychiatric disorders. And in fact, if you look at this distribution, This is how common things are, from very rare up to present in half the chromosomes in a population. This is the odds ratio conferred. And then each of these points is one SNP per trait association. The vast majority of things are in these two clusters, things that are common, things of low effect. In fact, the median across all of these is around 1.2. So a relatively small impact and risk that only 10% actually exceed 2. So again, typically across all areas of medicine, all complex traits, things tend to be relatively subtle and relatively common. This is a widely replicated finding. And here is when I make my brief segue into the Cytogenomics Consortium that Joel mentioned before, the PGC. This is a graph of papers written by people in the PGC. So as you can see, we're predominantly an East Coast US, Western Europe phenomenon with an interesting contribution of people from Australia. The PGC has cranked out 153 papers as of 2 months ago, and as you can see, last year was a banner year, and this year is about as big as every other year to date. So we're looking forward, and this is terms of impact. On this metric, each of those points is a PGC study, and this is an NIH metric from iCite. This is the median citation for NIH grants in the same area. The median PGC paper scores in just under the 90th percentile. And one of the highest cited papers that the NIH has ever funded is the 2014 schizophrenia paper in Nature. That's been our champ so far. Here's where we are in studies of common variation for psychiatric disorders, the ones that the PGC studies. So this graph, log 10 number of cases in the analysis. This is the number of independent and highly significant associations. So we have OCD, we have Tourette's, which again are really too small to really make any conclusions, alcohol dependence, anorexia, ADHD, autism, PTSD, and bipolar disorder. Here we go from, when I made this graph, this was what was published to what was in pre-publication step. This is schizophrenia, what's been published, what we're working on right now, and this is the graph for depression. As you can see, in general, the more we do, the more we find. Things that have higher heritability tend to be a bit easier, but really, in the end, it's a numbers game. One of the great things about depression, for instance, is that there's very large studies now that have been done. The UK Biobank has 500,000 people. There's a bunch of super large studies that people are doing, and depression is common. As long as it's been assessed in a reasonable way, we'll have a chance to increase. The leaders, the depression group, are hoping to have the next analysis from the PGC to have 1 million cases with depression. And so we'll see where that takes us. Okay, if I had to make a snapshot right now of where things are, the data here are for schizophrenia mostly. Here's what it would be. First, the inset shows rare variation, and I've got intellectual disability, autism, and schizophrenia. The colored parts mean that if you were to do a genomic full press, the typical assessment of rare variation and common variation, this is what you would find. So for the yield for severe intellectual disability is probably up to 2/3. Depends, that's not fixed and final, but it's going to be something relatively high. For autism, if you actually go for it, you basically find that you can potentially explain autism in about maybe a quarter, maybe high teens, maybe mid-teens. People argue, but something like that, a sizable subset. For schizophrenia, it's only a couple percent. Okay? So again, the theme that I mentioned before I think is evident here. The things that have the earliest onset and the greatest degree of severity tend to have the most findings from a genetic perspective, if you're looking for things that are rare and relatively strong effect. And I've tried to illustrate this on this graph, where this is for schizophrenia, where we have log 10 of how frequent the risk allele is. That goes from super rare all the way up to super common, and this is the risk conferred. What we see here, there's the single exome finding. These are 22q11 and the significant copy number variants. And then we also see down here, that's where the GWAS findings are. And as you can appreciate, there's sort of this interesting relationship between this, that the rarer things become, the stronger the effects. The more common they are, the lower the effects. And I'm going to argue that each of these has both therapeutic and clinical relevance, both classes here. Now, one of the things that has bedeviled us when somebody made this, the unfortunate terminology of, quote, missing heritability, it's been the thing that's bedeviled the field for a long time. People have wondered, where is it, what is it, help. that everything is useful because it's not there. The basic concept, though, is we know now that for basically every complex disease or trait, estimates of the twin or pedigree heritability that we determine from large population-based studies, a twin study that comes up with the heritability. Heritability of schizophrenia is 65%, say. But we can also measure heritability directly from the genetic variants we measure, and the two numbers are actually pretty far apart. So usually the pedigree heritability is about twice the SNP heritability, what we determine empirically from genetic data. This has led to huge amounts of teeth gnashing and lots of silly arguments, et cetera. And we see it for schizophrenia, we see it for height, body mass index, type. And the question for this is why? Is this something that is relevant and is it important? I've argued that it isn't important because basically we're looking for biological toeholds, and whether we explain heritability or not actually hasn't been part of the picture. So to go back to this, so essentially a version of this graph has actually been in the literature for at least 11 years. So essentially you've got findings over here that are potent but rare, and there are few of them. You have findings over here that are weak, common, and quite numerous, and then you have a gap here. And that gap is merely technological. This is an area that's actually hard to get at with either the two technologies that we do to get to either side. And there was a recent study from Peter Vischer and colleagues where they actually filled in that gap. What they did was, this paper's in bioRxiv, and it's one of the most interesting papers I think Peter's written in a long time. But basically, 21,000 people that had whole genome sequencing from the top med study. Whole genome sequencing is when you resequence the entire genome at relatively high depth. Every base was covered by about 38 reads on average. So that is the way to really get at pretty much all the variation in all of the genome. And what they found was, when they did this, they found the heritability they found from looking at the genetic data was almost identical to what you would expect from the pedigree data. So it's not missing heritability, it's misplaced heritability. And where did it lie? Well, it basically was exactly where you'd expect. In things that were relatively uncommon, had relatively stronger effect, and because there was no correlation, low LD, with other genetic variants, these are things that could not be picked up otherwise. So we have a simple resolution to a complicated problem. Okay, in summary, the genetic architecture is the term we use to describe just all of the genetic influence in schizophrenia. How many genes? Well, and this applies to basically every other psychiatric trait as well. Our estimates are that it's probably high hundreds numbers of genes. Most of the variation that contributes risk is common. Some is uncommon, some is rare. The effect sizes tend to be weak, some are stronger, but again, we need to get more firmly into that gap that I mentioned before, and we don't have studies of that yet. Generally, these things act in an additive manner, not in a recessive or dominant. We certainly suspect gene-gene and gene-environment interactions, but largely these have not been proven, and the studies have been a bit murky. And I also think that what we need to do next is now pretty clear. And if you're interested in the full details, we had a full-length review on that in the American Journal a couple years ago. Okay, but one of the things that's popped up is stuff like this. This is the genetic findings for approximately 2 million base pair region on chromosome 1. This is schizophrenia, the red, 1, 2, 3 associations that are all independent statistically, 2 intellectual disability associations, and an ADHD association. And I have never seen any theory even to explain what's going on here. How is it that genetic variation in this region actually increases risk for these disorders? There's literally no biology. We're just at the beginning to even articulate the problem to try and figure out what is going on here. And again, this is an example where what we found in these genetic studies have been different than what people would have anticipated. And one of the issues is that a lot of the methods we have are based on linkage disequilibrium, the tendency of variants in the human genome to be correlated with one another because we're a relatively young species. But that is great on basically if you're looking at the genome at gigabase scale, but it really breaks down when you get down to the megabase or even the kilobase scale. So next, I'm going to throw in something that's a bit cross-cutting between diagnostic and therapeutic, really the biological implications. What really is going on? And one of the issues we have is we find a GWAS locus. This is a genetic region that has multiple variants that are associated with schizophrenia, for example. These things tend to be about 100,000 bases. They tend to have many different variants in them. They're all highly significant, and it's really difficult to disentangle. Here's an example of the gene CACNA1C, which is one of the most robust findings actually in all of psychiatry. First came into the literature for bipolar disorder 11 years ago, and as you can see, here's the gene, and there's an intronic region that actually has relatively strong signal. We did a study a few years later for schizophrenia. The p-value went from, this is 10 to the minus, 5 times 10 to the minus 8. It went to 10 to the minus 12. In the 2014 schizophrenia paper from the PGC, it went up by 6 logs to 10 to the minus 18. And the unpublished stuff that's in prep right now, it went up another 5 logs. So the p-value here is 10 to the minus 23rd. That's something like 15 logs more significant than the bar we usually do. This is a pretty confident thing. I mean, it replicates everywhere. It's super strong. The problem is the information content actually hasn't changed. This is the region that was associated, and it's basically the same region that's associated now. So it's a lot more significant, but there's been no fine mapping. There's been no narrowing of the interval. It's the same, approximately the same 100, 116,000 base pair region all along. What on earth is going on here? And the late Pamela Sklar and a group had a paper where they actually used a technique to look at how parts of the genome touch each other in brain nuclei. And they made the case that there's actually a variant in this region which physically connects with the promoter to influence the regulation of this gene. That's the hypothesis. And effectively what we need to do is we need to do that kind of analysis at large scale. So they used a technique here which was essentially, we're just going to look at one area and see what it touches with. It's possible to sort of scale that in a way to look at all regions versus all regions. And here I'm going to tell you a tale of 2 heat maps. The one on this side has to do with linkage disequilibrium. So essentially, this is the correlation of the tendency for a genetic variant here to be strongly correlated with the genetic variant here. This one's correlated with that one, et cetera. So we've known for a long time that the human genome breaks into blocks, okay? And this is one type of blockiness. And this blockiness comes from recombination patterns. It comes from the evolutionary and factors in our parents above us. Now, the thing that's interesting about this is this phenomenon is identical in every tissue in your body. If I were to get a DNA sample from blood versus a skin cell versus a brain cell, we would more or less see the same thing if we were to do this. And it's a double-edged sword. It's the thing that allows GWAS to work in the first place, but once we get down to a narrow enough interval, it becomes really, really hard to separate it. It becomes very difficult to get beyond the fact that these regions are all just densely intercorrelated. And that brings us to the second one. And this is another type of heat map. The genome goes in this direction here, and here it goes on the diagonal. And basically, this is a high C contact matrix. This high C is a technique to look at, to identify the regions that are touching each other in brain nuclei. Okay? So this is really a 3D thing. So the nucleus is organized in a very complicated way, and we can identify the regions that are actually physically proximal in the nuclei in a sample. And this is another type of blockiness. And as you can see, these things tend to sort of, so in this region here, for instance, these regions all tend to have pretty high contact, but there tend to be few contacts between this region and this region, et cetera. So this is something that the technology is complicated and expensive, but it's doable. But the idea is this one, the LD-based one, represents mostly recombinational practices that happen in the making of us, as well as population selection pressure. That one is a functional snapshot of what's happening in one tissue at one point in time. So it's really a rather different thing. It's adding a functional overlay. And when you do this at scale, and this is a paper we've submitted, so basically we've contrasted ways to connect the GWAS signal to different genes. The way that we always do it is first by location. Here's our GWAS locus. Let's identify the genes that are in there. The second is to use this high C method that I mentioned. In other words, to look at things that contact. So we've got a region that we think is interesting from genetics, but we look to see the regions that it connects to in 3D in the brain. It could be in the region. It could be relatively far from the region. Another way to do this is using an EQTL. This is an instance where you have an expression relationship. So you have a segment of DNA that's implicated by GWAS. There's a genetic variant here that relates to the expression of a gene that's in the locus or far away. Okay, so these are 3 different methods to connect genetics findings to different genes. And when you compare and contrast them, essentially we see location is great in the sense that it's specific. If a gene is in a locus, it probably has one of the other types of evidence too. But it's not sensitive. There's a lot of things that are outside. So for example, this is the pie for location, and as you can see, all but 34 of these are actually implicated by other things too. But whenever you look at the high C information, you get a lot more genes, and a lot more genes if you look at the EQTL evidence as well. They tend to overlap, but at the same time, these are the alternative ways to overlay functional genomic data on top of genetic data to try and illustrate what's happening. And it's possible to, this is no secret, so ENCODE roadmap has generated a ton of data, not a lot that's actually relevant at all for brains. PsycENCODE was funded by the NIMH to actually do an ENCODE-like project that's brain-based and psychiatric case control based. They had a stack of like eight or 10 papers in Science last December, and they're embarking on their next level, which is going from bulk tissue to single cell work, which will be really fascinating to see how it goes. The Common Mind Consortium has done this, GTEx has done this, Cell Atlas is all about this, lots of single studies. The basic concept is that when we overlay the functional data on top of the genetic data, it actually tends to illuminate what's going on. Okay, diagnostic implications of all this. So I'm going to talk about a few things first that has to do with technological readiness. So I'm switching from just covering the findings to covering how they might be used diagnostically and therapeutically. Okay, diagnostically, I think it's pretty clear that this is something that's ready for right now. For childhood onset and severe neuropsychiatric disorders, in many institutions, this is in fact the standard of care. If a kid comes in with severe ID or severe early onset autism, they tend to get evaluated in many academic centers these days. The yield for moderate to severe intellectual disability will be up to about 2/3. Autism, maybe 1 in 6. OCD and Tourette's, I think we need to see more data on, but it's potentially promising. And essentially what you do is you do whole genome sequencing of both parents and the child. And then the output is, from a genetic perspective, are large structural variants, copy number variants, de novo variation in important genes that is absent in the parents but present in the child, and third, rare recessives, where each parent has one copy of a recessive and the child has two. And the diagnostic utility here is first, you get the correct diagnosis. People talk about the diagnostic odyssey that these children have, where often they will see dozens of specialists, all of whom are a bit stumped until somebody says, oh, wait a minute, we should check the genetics, and they say, okay, they have this syndrome, and all of this other stuff makes sense. And this is something which is just a huge burden and huge expense for parents as well. I think especially for when the parents are in reproductive ages, there are family planning implications often too. Many of these variants are de novo and unlikely to occur a second time. Some are inherited, and many parents would want to know that. I also think this is important from the perspective of service provision as well as support connections. So there's Facebook pages for many rare genetic syndromes that bring parents together, that share tips and secrets and all sorts of stuff. And largely, that's actually quite a good thing. In addition, from a physician perspective, many of these conditions are multi-system disorders. And it's not just like you can take care of the psychiatric manifestations. There's often cardiac and renal and hematological implications for all these things that need to be considered. This is mostly for the second section, but I'm going to measure it here now about the therapeutic utility. There are a few of these conditions that actually do have a therapy right now. And I know from the people I talk to is that many of these are actually the model for pharma company, new biotech startups that are actually trying to develop a specific therapy for a rare condition. And so I think if that's the case, it's likely that some of these will actually lead to useful findings at some point in the future. But you have to know which of your patients actually have them for them to be eventually eligible for these therapeutics as they come around. Something that I think is ready for a clinical trial are copy number variants in psychosis. If we were to take a sample of 100 people with schizophrenia, we would probably find 2 or 3 of them that would have a large known pathogenic copy number variant. If you actually increase the severity level, if you look at treatment-resistant, super chronic people, bad outcome people, however you define it, the yield is probably going to be higher. And The technology you would apply here is relatively straightforward. Array-CGH costs a couple $100 per person, and it would give you whether or not this person has one of the large known structural variants. Diagnostic utility is, again, getting at the correct diagnosis. For some people, family planning, service provision, support connections can be relevant. Again, these are multi-system disorders, and identifying and ameliorating the effects of other organ systems that are infected can actually be super important. And again, therapeutic utility. But I think this is not something which is as straightforward as the application to younger kids with severe early onset disorders. I think there's a number of pros and cons, and at present we're just having an awful lot of relatively wordy and quite articulate and informed, but relatively wordy arguments about this. I think it's time for us to put it to the test. We need data. So I think we should do a study where we get 1,000 consecutive patients, we do this in a consistent way, and we assess in a very clear and incisive way what are the impacts, positive and negative. What's the impact on the person, the family, on the clinical team? Does it harm anyone? What are the risks? Are there unintended effects? What are the costs overall? I mean, there's a couple of disaster, awful scenarios I can envision from this. I don't think they'll eventuate very often, but that's my opinion. I don't have data to back that up. I think we need to do the study. And I also think currently, I think for selected patients, we should consider doing a genetic workup right now for the reasons I've mentioned before. So for example, I would argue that we've all treated people with pathogenic copy number variants in the past. We just didn't know it. And some of these things can be super subtle. I had a woman that I knew had 22q11 deletion syndrome because it had been detected at birth when she had a VSD repaired. But you could not have picked this woman out of a crowd. She was just, her facies, her appearance was essentially identical to other people of her particular age and ancestral bracket. The flags that people typically talk about, premorbid low intelligence, developmental delay, congenital malformations, dysmorphic features. And I also think it's important to sort of always throw in the intuition of experienced clinicians. Sometimes, a lot of what machine learning things do, for example, is essentially pattern recognition. I like the pattern recognition of smart, informed, experienced clinicians. That's also a good thing too. And people are developing apps for this, where you can take a photograph of a face and get a yes/no flag as to whether this person has a higher chance or sufficiently high chance of having a genetic finding. So we've got some experience with this. So I've been doing a study in Pennsylvania with Rick Josiason and Marty Farrell, funded by Lundbeck for some time now. We have a sample of 500 people that really have ultra treatment-resistant schizophrenia. The first person in the study was hospitalized before he was born. He's been actively treated the whole time. Nothing works. He's as crazy today as he would have been 50 years ago. And what we've been finding on average is we have a relatively high prevalence of large copy number variants. We've been finding people that have 2 copy number variants. We've been finding people with a lot of frank neurological disorder that has either progressed over time or manifested psychiatrically. We had a case report in the American Journal of this exceptionally unlucky man who clearly had 22q11. If you go back and look at his developmental history, it's obvious. Developmental delay, ID, he had a bunch of congenital features fixed when he was growing up, et cetera. Clearly had it. He was treatment resistant from the very start. He was on clozapine after only 3 months of illness. And he was hospitalized for most of his life, and then he began to get worse, a lot worse, in his mid-30s. And as it turns out, he actually had Huntington's. And by the time we saw him at age 41, he was basically completely demented with no language whatsoever. This is sort of a 1 in 40 million occurrence. And obviously, the diagnosis of this made absolutely no difference to the treatment of this individual. But it did make a difference from his mom. She said that essentially, you can read the whole paper. She wrote a really nice addition to the case report. But her perspective was, as a mother, she had a huge amount of guilt about what had happened to her child. And seeing the genetic diagnosis made her realize effectively that there was nothing she could have done. And for her, that was actually exceptionally helpful for closure. So there can be benefits of doing this, which are actually above and beyond the benefit to patient, or the medical benefit. This is the second case we're writing right now. This is a woman who had an absolutely terrible upbringing, highly treatment-resistant psychosis, developmental delay, epilepsy, and we found that she has a 15q11 deletion. The resolution here stinks, but essentially this is about a 20-year treatment history of medicines that she's received. And so these are antidepressants, antipsychotics, lithium. I think this is all so antidepressants, antipsychotics. And essentially, and each slice going along is a week. And so I actually calculated that this woman had consumed 1.2 kilos of clozapine over time. And literally nothing worked. In fact, by the time we saw her, she had been on one-to-one for suicidality for about 2 years. Essentially, every time they weren't within arm's reach of her, she would actually try to do something to harm herself, and occasionally had actually injured herself fairly badly. So knowledge of this copy number variant did not itself make a direct difference to her treatment. However, at the same time, she had a couple of really determined and clinicians that were unafraid to roll up their sleeves to get in there and work hard. And it actually made a big difference. They were in the process of reconceptualizing her case as basically, instead of treating her as somebody who was willfully doing this, it was more, they started to put her more in an intellectually disabled category. And they basically stepped down their interventions to things that were really basic, really straightforward, and very, very stepwise. And that actually helped make a big difference with her. It also helped with nursing staff, because every time you try to change treatment, especially in where this woman was, it was hard. She'd been there a long time. They didn't want to do anything different. So the knowledge of this, they told me, actually helped confirm and support what they were trying to do clinically. And at this point, she's actually been off one-on-one for a long time. She's actually being prepped for discharge. So in this instance, it had some benefit, but it certainly wasn't direct. Okay. Another thing that we need to start thinking about, switching now from rare variation to common variation, is the nosological implications. And it's relatively easy for us to come up with heat maps like this. Essentially what we're seeing here is these are genetic findings from a whole range of conditions, cross-correlated. This is essentially a correlation. And as you can see, we find clusters. And I'll explain these clusters in a second, where we essentially find that some of these things are super highly correlated in a way that was probably unsuspected. So if nosology is indeed carving nature at the joints, what do the genetic data tell us? So we use something called LD score regression to estimate the genetic correlation between two traits. To what extent does the genetic basis for schizophrenia and bipolar disorder overlap, or schizophrenia and major depression, or schizophrenia and autism, et cetera? And what we find is that essentially the genetic basis for essentially all of these overlap strongly. It's really different from neurological disorders, where it tends to be relatively clean. But there's just a huge degree of overlap between the fundamental basis of these conditions, all of them. So again, this is probably what you would expect when you have a syndromic condition without a defined biomarker or a pathophysiologically based diagnostic schema, unlike neurology, for instance, most things in neurology. It's also highly consistent with clinical experience, epidemiology, and genetic epidemiology. But it really does favor the lumper category over the splitter. We probably have split things out too far, at least from a genetic basis. Maybe the splitting is useful clinically, but from an etiological basis, perhaps not. And the clusters that we see is that we find that schizophrenia and bipolar disorder have a relatively strong and consistent, major depression and the personality trait of neuroticism, ADHD, smoking, and childhood IQ. And the one that is really interesting is anorexia essentially has the genes, the genetic variants that increase risk for anorexia are associated with lower body mass and with sort of healthier metabolic traits, which is something that the group that's working on this is going deep on. And then finally, we find stuff like, if we look at the genetic correlation between bipolar 1 and schizophrenia, that tends to be stronger than the correlation between bipolar 2 and major depression, suggesting that the inclusion of bipolar 2 with bipolar 1 might in fact, even if potentially useful clinically, might not be the right thing to do from an etiological perspective. And this is something that I think we have to take a really careful look at. I'm not proposing that we rewrite the nosology, but we need to go back and revisit some of this stuff. Because it may be, there's also a strong correlation between anorexia and PTSD with schizophrenia. Now, we've sort of given, if you think about it, people with both of these disorders have beliefs or sensory experiences that are actually quite distinctive and to some extent could be considered hallucinations and or delusions. Clinically, we give them a pass. We remove them. And Joel had a really interesting paper that's in press, right? The PTSD one, okay. That essentially has, I definitely recommend reading it, but it basically makes this case. Maybe we need to go back and revisit that to understand whether there are diagnostic and or therapeutic implications. So again, that's one of the things this is useful for. And I should also mention that the rare variant findings of all types are similarly nonspecific. We've not been able to find, like an autism-only hit. It tends to be autism plus ID plus developmental delay plus other things. So it's not as straightforward as we would have hoped. Okay. Another type of common variation is something called a genetic risk score. A genetic risk score is essentially the summation of all the genetic variants across the genome into a single number, 1 degree of freedom, normally distributed. It's great. But essentially it captures the genetic liability to disease. And this is something, so it's a direct quantitative measure. We call these genetic risk scores, GRS or PRS, polygenic risk scores. It's probably the best biomarker we actually have in psychiatry. It's stable. There's under most models I can think of, there's no issues with reverse causation under like most biomarkers we measure. It's been widely replicated and it costs about $30 a person. And these are findings for coronary artery disease, where if you take the genetic risk score for coronary artery disease in a population sample, break it into centiles, you actually see this interesting march from low to high, where this is the highest risk, the lowest risk, and the odds ratio can get up to about 10. We found something similar for schizophrenia. Here we broke into deciles. This is the lowest decile. This is the risk in the second versus the first, et cetera. And as you can see, the risk climbs up in a very sharp way. And the odds ratio appears 10 or 15. Now, to make it a little more clear, I've taken the results from the green graph here and plotted it here. And what I've shown basically is that this is the schizophrenia genetic risk score, and determined from an external sample, and this is as a z-score. So this is a 1 standard deviation increase, 2 standard deviations, et cetera. The blue curve is for controls, and the red curve is for cases. This shows the mean for the 2 samples. So the mean difference here is hugely significant, 1 to the minus 200 or something. It's way, way, way beyond chance. But as you can see, there's considerable overlap between the 2 curves. This is effectively kind of like height in men and women. So the mean difference is massive, but it's not diagnostic because there's short guys and a lot of tall women. And but basically, when you get out to the curves, you really do start to see that out to the extremes. You do start to see quite differences. The proportion of humans under the red curve versus the blue curve at this point is really quite different. And what I've done here is I've taken the same data, and this is proportion, and this is the decile again. So these are people in the lowest decile, controls and cases, and the highest decile, controls and cases. And there's 2 real interesting things there. The first is in the top decile, we have controls that have inherited a lot of schizophrenia risk alleles and yet are unaffected. They do not have the clinical condition. We should ask why. What protective effects do they have? Do they actually have something that's akin, but it's not quite enough? Are these just sort of super paranoid people, for instance, but they've never been hospitalized, never been treated, whatever? And in the lower part, we have cases who have actually inherited conspicuously few, but you have the clinical disorder. Is this a misdiagnosis? Are these people that have had a massive environmental event that increase risk? Are these people who actually have been exposed to massive amounts of deprivation? Whatever. There's a number of things that we could look at. And again, from an investigative perspective, this is something we can do relatively routinely now at the point of doing $30 a sample. And if you do the power calculations, if you were to have a clinical sample of, where you've measured some phenotype really, really carefully of 300, 350, 400 people, you could add in this information at a cost of $30 a person, and the power calculations would indicate that if there's something reasonable there, you probably would find it. And so effectively, what we're doing now is instead of assessing, this is something that we advocate the use for in clinical samples to try and see whether it can help us get a handle on heterogeneity. Okay. I think something that we need to do, we actually need a whole series of studies for. And let me, before I describe what I think needs to happen, here's the goal effect. One of my colleagues in Stockholm did the Stockholm 3 study, and this was for prostate cancer, for high PSA levels. This was in Lancet Oncology 4 years ago, and in fact, this algorithm is now being used clinically in Stockholm. So essentially, it's to try and figure out clinically what do you do with men with high PSA levels. And so effectively what they did was they came up with a prostate cancer genetic risk score. They added to that some clinical data, age, past history, that kind of thing, just obvious things, and also some protein biomarkers. And using those data, they were able to construct an algorithm to identify 25% of men that they could, with high PSAs, that they could completely leave alone and do no further investigation, no biopsy, et cetera, and yet not miss a single important cancer. And given the prevalence of high PSA, this is something which has massive public health and financial impact. That's the kind of thing I think we should think about whether we can actually prove that this is something that we can do. Okay, like prostate cancer and high PSAs, all psychiatric disorders that we see clinically are heterogeneous. Indexing heterogeneity, of course, has been a major theme in biological psychiatry for several generations of research now. The question is, can we use common genetic variants to define etiological subtypes? Now, there are some studies that have looked at this, of course. Most tend to be relatively small, a bit too ad hoc, and I actually think we need a robust agenda that has super large sample sizes and a high degree of rigor so we can understand which of this might actually be clinically important. Okay, and then in the third part of this, I'll talk about the therapeutic implications. All right, so first of all, and this is my tip to aspiring psychiatric trainees, we actually need experts in this area. And effectively what I'm going to argue for is that we've got all these copy number variants that have been robustly associated with various psychiatric presentations. ID, DD, autism, schizophrenia in particular. But most of the literature that is focused on this has really been just about documenting the co-occurrence. We don't have data right now to guide therapeutic choices. What should we do, for instance, for that, for an adult with a 16p11 dupe and a psychotic disorder? What are the treatments that they should get or not get? For example, there's some people that say that people with 22q11 deletion should not get clozapine unless they're covered with an anticonvulsant because of risk of seizures, that kind of thing. These are effectively orphan diseases, given the prevalence of the combinations, and large systematic studies would be optimal, but they're going to take forever, and there'll be a global effort, they'll be expensive, et cetera. But yet, we really do need clinical guidance. So in this paper that Mike Owen and I wrote, we basically called for something akin to clinical crowdsourcing. Basically, the longitudinal follow-up of people with these combinations to write careful case reports that are clinically informative to help guide other individuals. The idea is that if we actually start to see a body of work like this, it might give us some ideas as to what works, what doesn't work, what might help, what to stay away from, et cetera. And eventually, it will also help build a workforce that can look at this carefully. And so again, I would argue that there's a niche here that needs to be done. Great clinicians, great psychiatrists and psychologists that really understand the disorders, that understand clinical genetics reasonably well, and also who read a lot and can come up with some best guesses and help increase the literature on this. Okay, turning to the therapeutic implications of common variation. All right, this has been tough, and part of what we've run into a lot is what Joel mentioned before. It isn't what people thought it would be. And I don't know about you guys, but for me, nature has never really cared much for what my expectations were for the way things are. I think we need to take schizophrenia and these disorders as they are and to figure them out. And so again, look, it's clear, the data I think are super clear that for pretty much all complex traits, there's hundreds of thousands of genes with small regulatory effects. I didn't talk about this yet, but there's definite overlap of these regions with pharmacologically derived targets. So if you look at the known list of receptors, for instance, that bind antipsychotics or antidepressants, they're definitely enriched for some of the things we're seeing. But we don't have any models for this. A lot of the work that we've done has been single target, single molecule causes, but that's not the reality. The reality is there's a lot of genes that come together in a complicated way. And so I think that the investigative ideas are this. First, can we identify the correct tissue? Now, this is using bulk tissue RNA-seq versus schizophrenia in this instance, and we see that it enriches in brain. This is mostly, there's no surprise here, right? And controls, and the specific regions that enrich for this analysis is dorsolateral prefrontal cortex and anterior cingulate. Again, no surprises, but a nice confirmation. The cool thing is that if we actually drill down into single cells, and the idea here is you look at genes that are specific that have expression in specific brain cell types and ask, is that small fraction of genes enriched for the schizophrenia genetic findings? And we did this in a paper that came out in Nature Genetics about a year ago. We evaluated 24 different cell types, and we found, and this is p-value going here, and we found that there were just a few, that actually, it wasn't all of them, it was just a few. Pyramidal neurons from somatosensory cortex and hippocampal CA1, medium spiny neurons from the striatum, and a couple of types of interneurons that popped up. And the reason I think this is true, or correct at least, is we've replicated it. There's a paper in bioRxiv where we used new data, some other new RNA-seq data set, three different ones actually, and we went at it everywhere we could. The results were basically confirmed. The positive controls made sense, and I think it's coming together into a reasonably coherent package. Moreover, one of our colleagues looked at the same region using open chromatin, a completely orthogonal technology, and more or less found the same thing. And again, all the positive controls made sense. And so I think the implications of this are pretty interesting. It does imply a network, and it implies that really what might be going on is at the level of single cells. What the GWAS results might be telling us might not be individual genes, but rather ensembles of genes that tend to be specifically expressed in certain places of the brain. And the advantage there, of course, is that tools exist right now for the manipulation of these cell types. And it also gives us guidance for if we want to make a cellular model or study one, which ones are probably the better ones to use. So again, I would argue that it may in fact make sense that for psychiatric disorders, the main output here is the cell type. And then for therapeutics, I think we can safely exclude a fuzzball like this, a densely interconnected, relatively small. It's probably something that's more like this, a large set of molecules that interact in a particular place in a particular way in the brain, and perhaps over development. I would argue that one of the things we need to do is to identify that network and also figure out how we can find a cellular model where we can tag it. It won't be one thing. Say we come up with 5 fluorophores that basically give us an idea of what's happening in this that's relevant to schizophrenia. And if we can do that, then we can actually take, if we identify that gene network, then we can take the power of modern pharmacotherapy or pharmacology at doing mass screening for this kind of thing to really start to walk through an intelligent way therapeutic effects. I don't think it'll be a single magic bullet. I think it'll be a magic shotgun. I think we'll probably need to actually evaluate combinational therapy. I don't see how we can do that in a practical way unless we actually have this idea, a network plus ways to tag the network. And then the final thing that we're doing a lot with common variation is we're interfacing with our consumers, the companies. So the PGC has had a couple of meetings already that were well attended and quite exciting. This one was reported in Biological Psychiatry. That was the one last year. And we're working with the NIMH. They're trying to get something spun up to look at how do we alter the outcome of the psychosis prodrome. Okay, so I also think finally that we actually need comprehensive studies of clinical outcomes. So this is something else which is investigative, but basically, can we use genetic risk scores to understand things that are important clinically? Again, this is a research investigative idea. What are the outcomes? If we actually use genetic risk scores to determine like subtypes of individuals, who has good outcome, who has bad outcome? What about transitions? People that initially have depression but have an increased risk of transition to bipolar disorder to schizophrenia, or poor outcome bipolar disorder. What about therapeutic stratification? Can we identify subsets of people for whom certain therapeutics would be better than others? It's all doable. Here's a paper that we just did recently with Karina in my Swedish group. Basically, we fit a model trying to predict treatment-resistant schizophrenia in 24,000, everyone in schizophrenia, everyone in Sweden with schizophrenia. And then once we had that model, we dropped down into the subset that we had genetic data on, and we basically found that treatment resistance was predicted by family history of schizophrenia and lower premorbid IQ, and the genetic data did not add anything in this instance. If you look at the graphs, though, it was sort of, so this is the impact of schizophrenia genetic risk score on this, and it was trending in the direction where it might make a difference, but our sample size was too small. So again, this is the kind of thing that I think we need to do, to carefully build models that are as detailed as we can with implicit replication, which we had in this, I'm just not showing it here, and also which basically are trying to sort of understand clinical phenomena that might be of help to individuals that see patients in a direct way. Okay, so basically, where are we right now? We know more for certain about psychiatric disorders than ever before. And I would argue that's actually pretty cool. But it's not something that we hear a lot. Most people tell me that the work we're doing has failed. Obviously, I disagree with that. And the reason I disagree is not my opinion, but rather because I think the data support the statement. So in terms of what's next, I can talk about this more if people want during the question section. But I think all that's left here is to thank you for your attention and to thank the cast of thousands that have made all this possible. So thank you very much for your time.